Welcome everybody, and thank you very much for joining. My name is Domenico Trico from Italy, and today it'll be my pleasure and honor to host this journal club of the EASD Academy together with Dr. Ilkesen, my co-host today. As usual, this journal club is organized in partnership with Diabetologia, the official journal of our beloved society. Let me briefly introduce you to Dr. Laura Corbin, who is a brilliant scientist from the University of Bristol, who has very kindly agreed to present us with the interesting findings of a recent study about the metabolomic signature of weight loss and remission in the direct trial. She earned her PhD at the University of Edinburgh in 2012, sorry. She already has quite a long experience in the generation and analysis of metabolomic and proteomic data. She's also an advocate of reproducible science, and she's actually an associate editor of Diabetologia. Many congratulations on these great achievements, Laura. Thank you again for joining us, and now I think the floor is yours.
Great, thank you very much. My background's actually in quantitative genetics. So the PhD that I did at University of Edinburgh was in quantitative genetics, actually in equine genetics. And that was around the time that the first SNP chip was introduced to horses. So yeah, a kind of strange background to start off with. But I started at Bristol as a senior research associate about 10 years ago. I've worked on a couple of major grants in that time, one developing methods for an approach called recall by genotype, and then the second one, which is the one that funded this work, looking at BMI and adiposity and the impact of that on health using metabolomics as intermediates. So yeah, I wanted to start by giving a little bit of background and kind of context for the work that I'll present today. So in our research group, which is headed up by Professor Nick Timpson, we're interested in dissecting the causal pathway between obesity or BMI, other measures of adiposity, and downstream disease. So whether that's cardiovascular, metabolic, cancer, and so on. And our general approach is one of triangulation. So that is to use a series of complementary study designs, all with different limitations and potential biases, to answer essentially the same question. And in this context, we're interested in using metabolomics, but also proteomics, and using those metabolites as intermediate phenotypes to help us unpick the metabolic consequences of overweight and obesity, and how these might be relevant to later disease. And in this way, I guess our approach is somewhat orthogonal to a multi-omics idea. So when you're thinking about multi-omics, you take a single data set or a single study, and you might evaluate genetics, proteins, metabolites, all in those same people. So that's a kind of multi-omics perspective. Whereas we take a single omics, so in this case, metabolomics, and then we measure it across lots of different studies and answer questions that way. Genetic epidemiology is a major focus for us. So cohort-based study designs, including Mendelian randomization and recall by genotype, are something that we do a lot of. But trials of weight loss interventions are also extremely valuable. So unlike what we see in cohorts, trials deliver substantial weight change over a relatively short period of time and enable both within individual comparisons, so we can take samples before and after the intervention, and also, if we're really lucky, across trial arm comparisons. So we might have a control arm and an intervention arm. And together, having that information together limits the influence of confounding and reverse causation on our analyses. So that's why we get quite excited about this kind of trials of weight loss interventions. And the work I'm going to speak about today is one of those clinical trial projects that I've worked on. And so we were really fortunate to be able to team up with some fantastic clinicians based at University of Glasgow and also Newcastle University, who were running a randomized control trial of a weight management program in patients with type 2 diabetes. And many of you might be familiar with DIRECT as a trial. So they've published quite a lot, including their main paper, which was several years ago now. And the program was built on the foundation of the Counterweight Plus program, And this involves patients being put on a very low calorie diet for a period of about 12 weeks, followed by a structured reintroduction of food, and then long-term support in terms of maintaining that healthy lifestyle. And the trial was rolled out across 49 primary care practices in Scotland and Tyneside, which is in Northern England, with whole practices being randomized to deliver either the intervention or the control, which is a kind of standard care. And in total, 298 patients were included in the final RCT analysis. And the primary outcomes of the trial were 15 kilograms of weight loss or more and remission of type 2 diabetes according to the criteria that they set. And then what we were able to do was to take serum samples that were collected at baseline, so before the start of the intervention, and again at 12 months after these 3 steps of the intervention. And these samples we sent for analysis by 2 commercial platforms, so one offered by Metabolome and another by Nightingale Health. And these approaches are complementary to one another. So the Metabolome platform is based on mass spec, and it delivers semi-quantitative data on over 1,000 metabolites from a single low volume sample. So you really get to see everything that's in there. And that includes lipids, amino acids, carbohydrates, but also, interestingly, you can get xenobiotics measured. So these are things like derivatives from drugs that people might be taking, medications. In contrast, the NMR approach, so the Nightingale approach, gives you accurate quantification. So it gives you millimole per liter amounts of around 180 molecules plus some ratios and other related measures. And this is quite lipids focused, but also includes some amino acids. And some of these are actually clinically validated tests. So I'm taking a slight diversion from the kind of diabetes and biology for a second, just to talk about pre-analytical steps. So as was said in the introduction, I do have a keen interest in kind of reproducible science. And from that point of view, I kind of tackled this during the process of doing this paper. So we sent 574 samples for analysis and received the kind of standard deliverables from Metabolome and Nightingale, which essentially is just an Excel file with our data in. So you just have all of the samples, all of the different metabolites, and you have this Excel file. And the first thing we wanted to do was simply understand these data, which sounds simple, but when you've got thousands of measures, it ends up being quite complicated. Wanted to characterize it and just understand those characteristics so that we knew when we got to the statistical modeling point that we were using methods that were appropriate for that data. But what we found was there was no real standard way of doing this in the literature, and that actually reporting of these pre-analytical steps was a little bit sketchy. You look at publications and not really know what they'd done to the data before they did their statistical analysis. And because we knew we were going to have quite a few data sets to process across our different study designs, we decided to build this kind of pre-analytical pipeline. And the rationale here was or the motivation was to have a single pipeline that would extract the data from the Excel file, it would summarize the data, and it would allow us to apply some quality control filters. We wanted it to be flexible so we could tailor our decisions about kind of filtering depending on the data set and depending on the study design and the data type. And then with this done, we would have a reproducible workflow. We'd also have a citable process essentially for our data preparation. And with help from David Hughes in my group, we've now been able to create this freely available R package, which will process metabolomics data, generate standard outputs, including a PDF report. There's an extract from that in the top right of the screen. And we were able to have a publication accompanying that in bioinformatics. So that was kind of a sideline of the project, but something that I was really committed to kind of delivering and that we've used over and over again since then. Okay, so back to the trial and the data itself. So having evaluated the properties of the data and performed some additional cleaning, I then set up this statistical kind of analysis plan. So I can come back to this slide because I know there's quite a lot of detail in here. But basically the primary analysis was a linear univariate linear regression model, and that's just evaluating the intervention effect on the metabolite. So comparing the metabolite level before and after in the 2 arms. I also ran a logistic regression model applied to the data after transforming it to a presence-absence phenotype. So that's explained on the right of this slide. So the thing with metabolomics data is, especially the mass spec data, it's picking up everything in the sample. So for example, if there's a drug derivative, it will pick that up obviously only in people who have taken that medication. And so there'll be a lot of missing data, but that missing data is actually really informative because it's not really missing, it's not there. So it's a null essentially. There's none of that metabolite in that sample. And so this logistic regression allows us to look at the presence versus the absence of metabolites in the samples. And so really wanted to include that because I think that can be, as we'll see, quite informative. And then the section in the middle, the exploratory analysis. So these are all the things that I was doing to try and understand, having done the analysis, which is actually the relatively easy bit, is looking for associations. But then when you get those associations, what do they mean? What do you do with them? And that's the kind of stuff in the middle. And I'll show some results that will explain that a bit more. Okay, so of the NMR metabolites that were tested, so these kind of about 230, I think 220, 230 NMR metabolites tested, around a quarter of those were associated with the intervention. And over half of those were showing an increase in response to treatment. And the strongest association was seen from glucose, and that makes a lot of sense. Around 12% of the mass spec derived metabolites were associated in the linear model. And then a further 19 of the less commonly detected metabolites had differential missingness by trial arm. So these are the things that are not always present, but when you do look at the presence, it's unbalanced across the different arms. And what I found really interesting was that we seem to be able to detect not just a weight loss signature, but also indications of sustained changes to diet and lifestyle. So remember, the 12-month samples are taken well after the very low calorie diet. So by 12 months, even those in the intervention group are on a kind of relatively normal diet, and they're getting advice about lifestyle and diet, but it's normal food. It's not a liquid diet anymore. And we could see signatures of changes in things like markers proposed for garlic intake and alcohol, for example. So clearly there were things that have been lasting change in these people. Also, reassuringly for us, the strongest association in the logistic regression model was metformin, which was depleted in the intervention arm at 12 months as compared to the control arm. So, the only people that really achieved diabetes remission were in the intervention arm, and we could see that difference by looking at the change in medication usage in those individuals. And we were able to look back at the clinical records and validate, clinical and then what was seen in the sample itself. But as I said before, we get these long lists of metabolites. So we ended up with some, close to 200 associated metabolites. And it's very difficult with those long lists to actually get at, what's important here. What does this mean for somebody's health? So I did a series of kind of post hoc analyses to try and explore what these associations meant. And firstly, I'm very aware that while I'm running these univariate analyses, none of these metabolites actually operate in isolation. So it's useful to consider the correlation structure between the metabolites, so the interrelationships and how they change and move together during the intervention. So for example, the most strongly associated mass spec derived metabolite was a metabolite putatively identified as erythronate. Now, this is not an obvious candidate. There wasn't much in the literature about it, so it didn't make a huge amount of sense. But when I then built a dendrogram and kind of to appreciate the relationship between this metabolite and other metabolites in the data set, realized that this metabolite was correlated with and so sat in a cluster alongside some other metabolites that were common sugar alcohols. And all of these had betas in the same direction as erythronate. And these are found in fruit and also in sweeteners. So this additional context, this association started to make a little more sense. And one of the interesting things here is when you receive the data from Metabolon, they provide these labels for the metabolites. So a super pathway and a sub pathway, and that's what's shown on the screen now. But it's not immediately obvious from the super pathway and the sub pathway classifications that these things are related. But then in our data set, they're correlated. So, that's why it's useful to build the tree and not, I a lot of people are doing kind of enrichment analyses based on super pathway or sub pathway, which is fine. But we were actually able to see correlations between molecules that didn't necessarily look like they were in the same class. Another obvious question, I guess, is to what extent is the metabolomic signature that we observe associated with the intervention due to weight loss itself? And it's really important to appreciate, given the design of this experiment, this question is actually, I think, probably impossible to answer completely, because weight change in this in the trial is completely correlated with the intervention, or almost completely correlated with intervention. And so then it becomes correlated with many aspects of the intervention. So weight change will be correlated with the changes to lifestyle, medication, and so on, all the other things that are happening. But we did start to have a look at this question by a relatively simple approach, which was just to add in the weight change into our primary model to give us the intervention effect adjusted for weight change. And in most cases, as you might imagine, the intervention effect on the metabolite was attenuated because some of that variation was ascribed to weight loss or weight change itself rather than the intervention effect. But there were a few exceptions, and those are the ones that are on the screen now in this list. And notably, omega-3 fatty acids are in the list, and this makes sense because these are essential fats. So the changes we see are most likely driven by sustained changes in diet, not by the actual weight loss itself. One other thing that we wanted to look at was how remission status might be involved. So everything I've done up to now has actually been ignoring the remission status. So it's been ignoring, if you like, one of the primary outcomes of the trial. It's simply been characterizing the intervention effect, lumping all those people in together, whether they achieved remission or not. So then we looked to see if there was actually an interaction between weight change and remission status for our metabolites. And these figures, we can see that in some cases, the change in metabolite level by weight loss category was different depending on whether patients achieved remission or not. So on the x-axis here, we've got quantiles of weight change. So Q1 is the most weight change, or the most weight loss, sorry. And then Q4 is actually a slight increase in weight. So there's those 4 quantiles of weight change. And then on the y-axis is the change in the metabolite. And the black dots are those people who did not achieve remission, and then the gray is people who did achieve remission. And this is the data combined from the 2 different arms of the trial. So it's everybody in here. But what you can see, especially on the left, is that regardless of how much weight was lost, the black dots, if we look at the people who didn't achieve remission, the level of the metabolite or the change in metabolite is pretty null. It doesn't really change very much. And it doesn't matter how much weight the people are losing, it just stays the same. But then if you look at the gray, which is the people who did achieve diabetes remission, you can see there is a step change. It's quite a linear change between the amount of weight that was lost and the change in that metabolite. Perhaps not surprisingly, the most striking interactions were seen for glycemic traits. So here we've got glucose on the right and 1,5-anhydroglucitol on the left. And this was actually a metabolite that at some point has been proposed as a useful marker for type 2 diabetes. So in some respects, it's kind of reflecting the diagnostic criteria itself. But what I found really interesting from this is that it seems like there's quite a lot of people who lost a substantial amount of weight but didn't return to a metabolically healthy state. So whilst weight loss may be required for the type 2 diabetes remission to happen, it's far from deterministic. So just because you lose weight doesn't mean to say that you can recover your glycemic profile. I also built a PCA plot using just the associated metabolites and explored the relationship of those PCs with some clinical biomarkers over the course of the intervention. So including indications of metabolic health. So that's that kind of the graph at the top. So these are things that you might be monitoring routinely in patients. And then biomarkers relevant for non-alcoholic fatty liver disease are shown at the bottom. And what we see is that these metabolites do appear to capture variation in these clinical indicators of health, even things like patients' quality of life measured by the EQ5D. So there is an overlap with the clinical kind of biomarkers that are already in use. And then finally, rather fortunately, when I was just finishing off this work, a meta-analysis of prospective type 2 diabetes was published and the metabolic footprint of prospective disease. So from this, I extracted summary relative risk estimates for incident diabetes and plotted these against my estimates for the intervention effect. So in this figure, the intervention effect is on the x-axis, and then the risk, the relative risk is on the y-axis from that incidence of diabetes paper. And from this, you can see quite nicely the effective reversal of many of the changes associated with disease development after the intervention. So those things that are kind of increasing as people are working towards the type 2 diabetes diagnosis are coming down again afterwards, and vice versa. And then we essentially come full circle. So the next step of this work is actually what I described at the very beginning of the talk, and that's to compare these results with those alternative characterizations of BMI effects. So like I said, a few slides ago, this is a great study design for generating a big change in weight. It's great because you've got people's measures before and after, and it's great because we've got that control arm. But at the end of the day, there's a lot of stuff going into that intervention and changes that are happening above and beyond the change in weight. And teasing those out from within a single study is quite difficult. So what we do is we bring multiple studies together. And so we have a fantastic PhD student, Maddie Smith, at the moment, who's currently conducting a similar analysis in a cohort of bariatric surgery patients. So then we can look at the metabolomic signature of a bariatric surgery event. Then we compare this to the signature of the dietary-based intervention. And what we take from this is where we see common effects, then these are telling us it's quite likely something to do with the weight loss itself, because that's the thing that's in common between these two interventions. Whereas we might also see intervention-specific effects. So for example, the omega-3 fatty acid signal is not evident from the surgery study, as far as I can see at the moment, but it is in this direct. So there's things like that which will be different, but the things we see in common, we take from that it's more likely to be a weight loss story. And we can also, bring in other characterizations of BMI. So we take phenotypic and genetic predictors and observations of BMI in cohort studies, and we can align all of those up together and get just those overlapping signals that we believe are driven by weight or BMI. We have another project. So on the right is a project looking at a surgery cohort again, but this time with some healthy population samples analyzed within the same experiment. So the idea behind this was to try and see whether, if we have data before and after bariatric surgery intervention, then is it the case that those patients, once they've been through that surgery and kind of come out the other side, do they move closer towards a kind of healthy population, or a kind of normal population, if you like, as compared to before the surgery? Or do they move in a completely different direction? Do they have a completely different kind of omic footprint that speaks to the intervention that they've been through? Okay, so just finally, just some thank yous. So huge thank you to all of our collaborators in Glasgow and Newcastle. It's a huge effort to put together a trial like that, and we just kind of were so fortunate to get those samples and to run those analyses. So we're really grateful to that, and to also all of the people who participated in that trial. Thanks to the team back in Bristol. It's a really fantastic group. So if anyone is interested in the work we do shout, because we're very happy to have visitors at any time, so do let me know. And then finally, thank you to everyone for listening. And I have to do this final little promo, which is that we're doing a Mendelian randomization conference we're hosting in June. So if anyone is interested, do go to the website and have a look. Thank you very much. Happy to take any questions.
So thank you very much, Laura, for your very nice presentation. I don't know whether we already have any questions from the audience. So we've got raised hands. Yeah, Geronimo Ailman.
Yeah,
Please.
thank you, Laura, for that great talk and very nice, especially analytical insights. So this is Geronimo from German Diabetes Center, Düsseldorf. I'm asking first a very simple question. Maybe I missed it. But can you simply tell me how was the overlap between the metabolome panel and the Nightingale panel? So just approximately, did you find the same metabolites increased or not?
Yeah, they're pretty good. We have done some work kind of comparing the two platforms. The sorry, they do tend to track each other quite well. I think probably the error is certainly different across the two, and we don't have units in Metabolon, so we tend to rank normal transform the Metabolon data. We rank normal transform both data sets, but obviously you can go back in Nightingale to millimoles per liter, but in Nightingale you can't. But yeah, the correspondence is pretty good, and we also have clinical measures for things like glucose and kind of total cholesterol and things like that. So we routinely kind of check as much to look for, you know, sample mix up and ID mismatches as anything else, but they do generally correspond quite well.
Okay, and if I can have a second question. So again, really regarding your analysis, so I really will read this metaboprep paper. This is something I researched for since months. And I've seen in your volcano plot that you showed in the beginning, there were a lot of data points on the baseline. So I assume, did you perform kind of imputation in that whole data set? Because actually, I know all these baseline values are often derived if you don't perform any imputation for missing data points or something like this. And I think here you have an issue maybe about missing some information if you don't imputate. So this is at least what I learned in my PhD time.
Yeah, so I've just flipped back to the plot to remind myself. The reason for those ones on the bottom is because that's a plot of after FDR correction, after Holm correction. So a bunch of them go to a p-value of 1, and then they sit on the baseline. Okay. So that's why that looks like that. So the alternative is we plot the raw p-value, which I don't think there's necessarily a standard way of doing it, but that would make that look better. It's also a bit messy because in that volcano plot, I could probably share it again actually, because another thing that's potentially a bit confusing is that normally when you rank normal transform, you see a much tidier volcano, right? You see like a bowl, if you like.
Yeah.
But I struggle with the RN. Once you've ranked normal transformed something, I think you really lose a lot of interpretation and scale. You know, you don't really know whether that's a big effect or a small effect. So when I did this volcano plot, I actually switched out the beta from the model for a log2 median fold change. And again, since then, I think in the group, we've kind of been like, oh, I don't know if that makes sense. So we've changed again. But for me, I was feeling like I want to know whether there's two metabolites where one's doubled and one's quadrupled, because that's a useful metric. But do it on the median instead of the mean, so that if there's any crazy outliers, once you go back to the raw data, it kind of makes sense. So that's why that volcano plot probably doesn't look like a typical one that you would see with that kind of bowl shape where you've ranked normal transformed, so the p-value and the betas become much more correlated.
Yeah.
Was that the whole question? I've gone off on one and I can't remember.
Yeah, that absolutely answers a lot of this question, but I will have a look in that.
Oh, imputation. Yeah, we don't impute. Well,
yeah.
I didn't impute. Okay. And we often have this debate in our group about imputation, because in some senses where you've got a little bit of data missing, you can impute very accurately, but you don't gain very much because you only ever had a little bit missing. Where you've got a lot missing, the error of that imputation is probably greater, and so the information gain is less. So I almost did that kind of two-pronged attack where I do the linear model for things, and that tends to be the primary result for things that don't have a lot of missing data. And then for the things that do have a lot, make the assumption, which is an assumption and maybe not always correct, that missing data is actually zeros, and then do the presence absence check. So look for differences in the proportion of missingness instead of looking at actual values. And it's quite interesting doing it that way, because for something like metformin, if you just do the linear model and ignore the missing data, you get almost the wrong answer, because you get increases of metformin in the intervention arm, but it's an increase in concentration. And the problem is all those missing values should be zeros. And so then if you actually put them in as zeros, you'd see a decrease. Or if you look at the differential missingness, you see a decrease. So metformin is quite a good kind of thought experiment to do it for. And I think the reason you see an increase in the intervention arm, if you just look at the levels and ignore the missingness, is because those people who are still on metformin after that intervention have probably got quite a severe, disease diagnosis. And so they may actually be on, have more metformin in their system than the others. So yeah.
Yeah, thank you a lot for that insight. I think this is always a huge bias in such a data set. Thank you.
So very, very good. I don't think we have other questions from the audience. So I will ask one. I think you already partially answered to this question, but from a mechanistic point of view, I'm interested in whether were you able to determine whether these variations in the metabolome after weight loss reflect changes in metabolic processes that can actually influence diabetes remission, or perhaps they are just a reflection of the improved metabolic health status as we expect following weight loss?
Yeah, I think it's a really critical question, but I kind of in my head, I'm like, how do you ever separate those two things? And like I said, with this, I think with it, from this study, we have a set of potential biomarkers for weight loss and maybe even for remission if we do a little bit more and start to separate that out. And as I think I showed in my slides, some of those associations actually just reflect the diagnostic criteria. So, if you split people up by achieved remission and didn't, then you just see that glucose, difference between the two groups. But then I think the fact that you see the metabolomic signature that we see maps so well against that kind of incident disease signature really makes me feel like actually, maybe there is some causal stuff going on there, because that incident risk it wasn't a metabolomic footprint of where is it? When I go to it, it's not a footprint of actual disease. It's a footprint of, oncoming disease. They're about to flip into disease. And so I think, I was really surprised. I never expected that, to look so kind of neat in terms of the reversal of many of those things. But obviously not all of the things are moving back. So that's another thing that we could look at is, what doesn't change, that was changing on the way into disease that hasn't changed on the way out.
And now you should have like the 5-year follow-up samples from the direct study. I just read the another paper published in Lancet Diabetes and Endocrinology with the 5-year data on subjects who actually remained into the study or those lost at follow-up. So it might be interesting also to see whether changes are associated with long-term diabetes remission.
Yeah, I'm not sure whether they have, I don't think they have samples at 5 years, but we could certainly track forward. So that's one of the things that we haven't done yet, which I'm quite interested in, but this is a relatively small data set, which is to look at baseline and see if there's anything at baseline that tells you whether those people are going to achieve remission or tells you, how well the intervention is going to work. But those kind of prediction analyses require quite a lot of power, and I just don't think that we probably don't have the power in this analysis to do that.
I may also ask a question. Meanwhile, it's a simple one. You're talking about individuals when you tell about the samples. I'm not sure if it is both female and male, like it both women and men in your data set.
Yeah.
And if so, is the percentage similar, women and men? And also, if it is the case, did you look into sex differences in your data, and did you see any interesting outcomes?
Yeah, so we didn't actually in this. So the model is adjusted for sex. So we're essentially looking at sex averaged effects in all of the stuff that I presented. I think it would be something that would be useful to look at. And actually we're doing, in our surgery cohort, we've now got proteomics data. And I started to look at the difference by sexes and look at kind of a sex interaction. So does it, do you see a difference in the change through the course of the intervention by sex? To be honest, we don't see a lot of signal with the protein, but it is quite a small, again, to detect those interactions, the power is a little bit low in these kind of studies, I think, where you're kind of in the hundreds. But it's definitely something that's interesting, especially because we know kind of fat deposition and stuff can be quite different in males and females. So it's not something we've done here, but yeah, something that we're kind of aware of and thinking about.
Thank you.
So in the meantime, I will ask a more like general question. It seems that you're really expert about this new omics technique and precision medicine. So which are the research skills and qualities that you see as necessary for the next generation of researchers in our field of diabetes and obesity? Because some of us, we have not been all trained on these research techniques, and I think this may become really important in the future.
Yeah, I mean, so I definitely have learned on the job. So a lot of the kind of what went into the formulation of the MetaboPrep R package was me and my team trying to understand the data, making decisions, then realizing they were stupid decisions and we actually should do it a different way. And I wouldn't necessarily say that we've got like the gold standard now, but certainly a lot of thought has gone into, handling the data and thinking about, what we're doing and the impact of that on our results. I think as data sets are becoming larger and more complex, I certainly think there's a need for researchers to have kind of to be able to handle this bioinformatically. So in terms of coding and all of that stuff, but also, in terms of statistical modeling. And I think that's definitely something that we're kind of learning as we go along. Something we don't do a lot in our group at the moment, but I think probably will start to do a bit more is the kind of machine learning. And I think it's easy to get that stuff wrong. And so, to have people who really understand those methods and how to apply them, and importantly, their limitations, I think is going to be really important. I think, as has always been the case, we can't be experts in everything. So it really is about bringing together a team of people, all with different kind of strengths. So, maybe we need to bring in bioinformaticians, statisticians who have no knowledge of, the biology that we're looking at, but actually can say, well, obviously, you could do this because this is just a multivariate problem that, people have already solved. You don't have to spend time doing that. You just need to apply those methods. But, working with people from different backgrounds is also challenging because you have different languages, which can mean things take a bit longer to get going. I think it's worth, trying to put together those kind of multidisciplinary teams.
Very nice. We have a question at the chat I can see from Agatha. She's asking what was especially interesting about the direct trial that you chose this one to run the analysis.
Well, as ever with these things, I think it was partly kind of serendipity that we, the PI on this project happened to come into contact with the guys at University of Glasgow and Newcastle Uni when they were setting this up, and realized that actually this is a really useful, as I said, it's a really useful example of where weight can change pretty drastically in a small space of time. And that's something we don't really see in cohort studies. People do spend quite a lot of time looking at BMI trajectories over the life course, but actually you don't tend to see sudden changes. It's incremental. And if you do see a sudden change, it's normally because there's been some kind of disease insult or something like that that's really triggered that change. So I think there's probably many trials that we could have worked with, to be honest. And I think, we're really keen on making use of the data that already exists. So where there are trials and they have got samples, we don't need to reinvent the wheel. We don't need to do a study again. These are the gold standard. And so if we can add in metabolomics, proteomics, whatever else to enrich these data sets, then, that's what we want to be doing.
So very nice. So we've got another question from Aram from the audience. And so the question is how the role of metabolomics be applied on localized visceral adipose tissue. And he or she said also, in particular, I refer to epicardial adipose tissue and its crosstalk with myocardium in arrhythmia and heart failure. Well, that's really specific, Aram, I think.
That's
So
really specific.
that's okay. So I don't know whether you can provide an answer on this. So, but please.
I mean, I can comment on it. I wouldn't necessarily say I can have an answer for it. So I think one of the important things to think about actually is all of the stuff that my group do is on circulating metabolites. And of course, you know, what's in the circulation doesn't always reflect exactly what's going on. Well, it certainly doesn't reflect what's going on in the tissues. It's a kind of strange accumulation of molecular events that you're capturing in the circulation. And so what's interesting is sometimes what you see in the circulation can seem confusing but then you realize that actually we see this with proteins, I think maybe more than metabolites, but what's circulating is more a reflection of what's no longer in the tissue. So it's a kind of interesting idea that you need to kind of always be mindful of the fact that this circulating levels may not correlate with levels in the tissue. I think you can do metabolomics on tissue samples, but this sounds like it wouldn't necessarily be a sample that you could obtain to run that on. So I know that Metabolon do, I'm pretty sure they do kind of, yeah, tissue-based analyses. We work closely in our group with some kind of cell-based, so all the work I do is dry lab, but we work closely with a wet lab group who are particularly focused on cancer, but they will do these kind of analyses sometimes on kind of cell-based assay level. But yeah, it's really hard to think about, you know, I'm not sure whether circulating metabolites is going to get you kind of the information you need for these more specific tissue-based questions.
I can also ask another one meanwhile. About the my question's about the metabolites that you find after the weight loss in the intervention group. So there are some, of course, that you expected to be changed, like the fatty acids were going down, as you said. But I was wondering, was there any metabolite that changed in the way that you didn't expect?
Or yeah, we have been a bit perplexed by we seem to have some sphingolipids going up, which wasn't exactly what we anticipated. And I think we're still thinking about that. We still don't have an answer for that. So yeah, I think there's always things that are unexpected.
Let me ask you another personal question, I'd say, because I mean, we don't see many early career researchers in the editorial board of prestigious journals such as Diabetologia. So I wonder, how did you get to this position, and how do you believe your role in the editorial board of Diabetologia has contributed towards your career, or has it been, I don't know, detrimental because of the time you all, of course, have to invest in it?
Yeah, I mean, it's kind of a tricky one. I'm not sure. I suppose I wouldn't necessarily say I was on the fast track in terms of progression. So I've always really enjoyed doing the hands-on stuff. So I've almost kind of kept myself in a position where I can still do some coding and still do some analysis, because I've been not wanting to necessarily get past that too quickly. And I'm, you know, as someone who works part-time and predominantly from home, and I've also had parental leave and stuff, you know, it is a bit of a slow process in many ways. But yeah, the role at Diabetologia it's been great. I mean, I've just been there a year now. I think mainly it came up because I was co-publishing with an associate editor who was already who was on the board and leaving, and so doing similar stuff. And so he's in my group, and so he was like, well, you know, I was doing a lot of similar stuff. For me, a major benefit is getting to interact with a new circle of researchers. So I'm not a diabetologist nor a clinician of any kind, and so, you know, it's kind of opened up my circle of, you know, associates, if you like, to people who are doing very relevant stuff, but stuff that I wouldn't necessarily come across otherwise. And like you say, it's a really highly regarded journal, so I'm really grateful for that, especially at a time when it feels to me like there's new journals kind of popping up all the time, and some of them we might think a little bit dubious in terms of their business models. I think everyone's now getting those emails that says we've got one more slot in our journal issue, please submit a paper, this kind of thing. So it's nice to know that you're kind of with a journal that's established and highly regarded. But it does take time, and I think I was reviewing a lot of papers before. I was probably reviewing a paper a month before. Now I'm editing a paper a month, so I think I've just reduced my reviewing and and replace that with the editorial work.
So I see. Thank you very much. Do we have the other questions from the audience? This might be your last chance, so don't be afraid of asking questions.
Meanwhile, I can ask another one, curiosity. For the last part of your presentation, you were talking about an ongoing study where you're gonna check the differences between the metabolites, which is changing based on the diet versus surgery. Then you will seek these differences. So how are you planning to do the analysis? Are you planning to pull all the data together and analyze, or separately and compare? I was just wondering about this methodology that you're gonna follow.
Yeah, so at the moment we restrict, that comparison step is really tricky. We're not doing it as well as I want to, really. So at the moment we just tend to compare lists. So that's what the scatter plot is essentially doing. It's just looking for overlapping signals. So where you have an association in one study, do you have an association in the other? Really simple. What I would like to do is to get to a stage where we can look more at the kind of profiles, so taking into account the interrelationships. And because obviously, you might have an association one study, you don't have an association in the other study, but another correlated marker is associated. So actually the signal is there, but doing that really simplistic one-to-one matching just doesn't take account of that. So I would like to do it more on a profile basis. We can't just stick the data sets together, especially, well, at least not for the mass spec data, because it's semi-quantitative. It's kind of normalized and scaled within the experiment. So if you try and put the two data sets together, they're kind of normalized to the median. They have a median of 1, so then they just can, if you take the normalized data, they just sit on top of each other and look identical. And if you take the not normalized, then you just get this massive structure because it's the experimental structure. And that was kind of what motivated the other study that I talked about, which was to actually analyze at the same time some healthy population samples, because that means we can do that then at the individual data level. So we can do that as a whole, because that was something we couldn't do. We're based, I think anyway, unless someone here can tell me a way of doing it, I think when we're comparing across those different studies that have been run through the lab at a different time, we're restricted to doing a kind of a summary statistics based comparison of some kind. So taking the outputs of the regressions and then comparing those.
Thank you so much.
Okay, so since it appears that there are no other questions from the audience, I'd like to thank again Laura for accepting our invitation. Thank you, Ilke, for hosting this talk with me, and of course thank you very much and thanks to all participants that I can see here in the chat. Let me just briefly invite you to attend our next Early Career Academy webinar, which interestingly will be hosted by Roy Taylor, the senior author of the direct trial. So we will hear again about this very, very interesting trial that has really changed our view or our clinical approach towards the diabetes remission by dietary interventions. The webinar will be held on April the 4th. So again, thank you and good evening, and see you next time.